My name is Nate Ju from California State University, Monterey Bay, and today I'll be talking about a cure that we've been doing in our intro biology lab on genome sequencing. It's the first semester of our general biology lab series, which focuses on molecular biology, cell biology, and physiology. Enrollment typically each semester fluctuates between about 80 to 140 students. We have a wide range of learning outcomes associated with this lab as we do both wet lab and computational work as part of this cure. The lab we refer to as the Genomics and Bioinformatics lab basically happens over the span of approximately 2 to 3 weeks. We are trying to target more towards 2 weeks. But the major outcome is that we want students to contribute to a novel discovery-based research project by making a material contribution to an active genome sequencing project. So that's sort of the main focus for what we are hoping to accomplish. And then there are a series of other learning outcomes that are complementary to that and also, we think, set a good basis for developing their skills as scientists. The workflow pre-COVID was pretty straightforward. The first week we did a DNA isolation protocol. The second week after which we took a DNA sample and our technical staff would then generate the library and sequence it on a MinION sequencer. And then week 2, we would just assess the results of our sequencing experiment. One of the unique things about the DNA isolation protocol is that we allow students to actually alter one component of the protocol to try to either reduce some sort of thermal or physical stress or contamination issue that might be associated with the DNA isolation process. This is all because it's done as sort of a competition among the different groups, and the team that can score the best in 3 different criteria, yield, size, and purity, will then be voted on by their peers, and that will be the thing that we'll actually sequence, the sample that we'll sequence for the sequencing. In the second week, we're just basically assessing those results, and they get some experience with a big part of this outcome being, can you identify repetitive regions of a sequence or encoding regions of a sequence? Post-COVID, things changed a little bit. Obviously, we couldn't do anything in the lab, so everything was made into video demos. Students didn't get to propose changes to the protocol thereafter. The data was provided by the instructors, and everything was done online. And we even changed the graded outcomes from a full report to just a results section. And you can see a little version of the sequencing lab that I set up at my house. We've got MinION sequencers over here and a little mini PCR that we use to do the library construction. Overall, we think it was pretty successful. Learning outcomes seem like they were mostly met. We saw positive outcomes in terms of what they felt like their analytical abilities were, motivation, self-efficacy, and overall, people had a general good opinion of it when I talked to the instructors and the students. I would say that there were areas for improvement, specifically in the area of student ownership, which is something that we would really like to be good at during a cure, but didn't actually have much of an impact on in the course of this experiment. And that probably had a lot to do with the fact that students weren't allowed to really invest in that protocol part that DNA isolation protocol part we normally do in week 1. And then we have some other things related to some of the writing outcomes that we're trying to emphasize in this class as well. So we're on track for fall 2020. We'll see what happens. We're trying to figure out some ways to maybe do some more group discussion work and build that ownership component, maybe having some discussions pre-lab and do things a little bit more asynchronously for some of the activities. For instance, they'll do all their video watching and all these other sort of background information exercises outside of class, and then we'll use the in-class time to do more discussive work. And then also trying to figure out how to take our outcomes and integrate them more into sort of this broader effort we've been doing to try to improve science writing outcomes. So I'm happy to answer any questions that you might have about our work, both our successes and our failures. And I just want to say thanks to all the other instructors and technical staff that have helped in executing this project over the years. Thank you.